I'm going to try to teach you a little bit of neuroscience. I know the audience has a wide expertise. And on the way, show you how we apply and try to change how our fields do open science. And eventually, I hope you will understand why we developed this platform we call OpenScope. But first, before I dive into this, I always like to start with our team slide. This work is the result of a lot of interconnected teams at the Allen Institute. We operate slightly differently than a traditional academic institutions. We have dedicated team to each part of the process where we acquire data. And so that means that it's really a matrix of people that created the work that I'm presenting today. So I'm only responsible maybe to like 0.1% of what I'm showing you today. But there are staff that are designing the experiment, technicians that are collecting the data, managers that are managing these teams, software engineers that are specialized into packaging this data, pushing it to the cloud, and monitoring this process. We also have like really important animal care teams. In addition, the OpenScope steering committee that is leading the platform, the observatory platform that we established, is composed of leaders in our community, including some that are here. Mackenzie Mattis, that has helped us shape this program over the years. And at the end, I'm going to talk about a community project that we have started this summer that is also led by a collection of professors and postdocs across the community. So please take this home. This is really teamwork. And that's actually quite important part of this presentation. Okay, so I want to talk about 4 things. The first one is try to answer this question: why neuroscience need more teamwork? The second one is what can we learn from other scientific fields? I think there are scientific fields that have pioneered open science for centuries. It's not a new movement. And then we talk about how we applied these practices in neuroscience and built the first platform we call the Allen Brain Observatory. And in the end, I'll introduce how we open this platform for the entire world use. Okay, and you are going to see the slides throughout my presentation. So I want to give credit to Saskia de Vries, who made these slides in the first place. And so I'm going to use this to showcase our progress towards open science over the years. So when I joined neuroscience in 2004, it was in Paris, this was our model. So you can see thick blue lines that's showing that most of these processes from designing a research question, building the experiments, collecting the data, analyzing it, publishing was close. So that meant that it was done in the realm of the lab, which is, it's great. That's how you push innovations. And then only at the very end, the science was pushed out. There was no open data policy, so you could request the author for access to the data, but that was the practice at the time. And the publication was most of the time paid access. So that was in 2004. And we've made some progress, but I'm going to use these slides to sort of highlight how it went for us. Before I dive into this, so what I would like to answer this question. So why system neuroscience needs more teamwork? You could argue it's been going great for a century now, and so why should we push for that? So I'm going to start by highlighting key challenges that our field is facing. So here you're looking at a recent publication from the Allen Institute. This is a mouse brain. A mouse brain is about the size of your thumb, so it's tiny. There's close to 100 million neurons. This is like about 1,000 times less than in the human brain. So it's a simpler system, but it's very tightly integrated. This is showing you the number. So at the Allen Institute, we took brains of mice, sliced it, and built an atlas that sort of is made using the genetic expression of genes across the brain areas and other factors like the anatomy and the density of cells. And we made this atlas a resource for the community. And so this has been really helpful because without a map, it's really hard to register findings from one lab to the other. And so this has a tremendous impact on our field. But this is showing you the number of areas that are involved in a mouse brain, which is a simpler brain than the human brain, in processing sensory information. So the challenge is quite high. And the problem is that those areas are all talking to each other. So it's not like a simple system. So in AI, most of the processing is done feed forward. I mean, there are models that are more complicated, but mammalian brains are tightly integrated. They are feed forward and feedback pathways everywhere. And we don't fully understand their roles. And so this is a domain of really, really important research. Now, you've seen that there's a lot of brain areas. What are inside those areas? So this is a publication that was released also from a collaboration with the Allen Institute and others last year. And they use an imaging technique called MERFISH. So we are sort of mapping out the presence of genes in the individual cells with high spatial resolution. And so at the top, you see a UMAP visualization. So again, it's using AI to visualize the distribution of classes of cells that are present in the mouse brain here. And they map the entire mouse brain with great details. They found a little more than 5,000 clusters of cells. And so now you see the challenge. Artificial intelligence system most of the time has one cell types. One cells that has great capability of learning new things, but in real brains we have many more of these, and so maybe but it's still, you might know that now AI systems are trying to train the models. They're so expensive to train in terms of energy. That takes a nuclear. They're now thinking about building nuclear power stations to feed these training centers. A human brain consumes much less energy. And maybe the answer to that lies in these specializations of cells for dedicated tasks. So there's tremendous interest in understanding what are all those cells doing. On the left here, you see excitatory cells that are and how they're distributed across the cortex. So the bottom is we call a coronal section. So you see the mouse brain from the front to the back. And I'm sure you start to notice there are layering patterns on the cortical tissue. It's the coloring. As this is showing you, there's some homogeneity in the distribution of cell types across the cortex. So this fuels a lot of excitement that there might be a computational principle across the cortex, which is the main tissue in human, that has really exploded evolutionary. On the right, this is the inhibitory neurons distribution. Again, most of the AI models do not have these. And so you see that they are also distributed in subclasses, and there's some sense of homogeneity across cortical areas. So understanding what all of these 5,000 cell types, classes, we don't really know which one are functionally relevant and which are like evolutionary dead ends but did not evolve further. There's a tremendous amount of interest in doing that. All right, and so at the Allen, we have started to map how the cells are connected. So here you see it's a summary. There's an entire publication on this. Where you're looking at the main classes of inhibitory neurons, parvalbumin, VIP, and SST cells connected to pyramidal cell, which is the excitatory cells of the cortex. And there are certain patterns of connections at play. So this month, and so the idea is that potentially we could extract computational principles on how the cortex receive and deal with incoming information. All right. And so now you've seen ideas of a map of how the areas are distributed. Now we understand a little bit the cell types and how they're distributed, and we have a sense of connections. So If you really want to understand how a real brain functions, you have to study it in vivo, in real, when the tissue is dealing with incoming information. So there's a lot of teams that are doing this. So I'm highlighting here a publication from Nick Steinmetz and his collaborators in England, UCL, and now he is in University of Washington. So this is a mouse is trained to do a very simple task. So it's looking at the three screens, and it has to move. So you see on the left or on the right, there's a little grating that appears, and the mouse has to turn a wheel under its feet, or left or right, depending on where the stimulus is. It's a very simple stimulus. And then they train a general linear model on the data. They recorded cells across the brain, one probe at the time, using electrical recordings. And they look at, okay, so do we have specialization of the areas, most areas involved in this task? And to some surprise, some more experienced professor might say we knew this all along, but most of the areas that we're recording from were involved in that simple task. That means, so you see on the top right, the percentage of neurons that are encoding the visual stimulus or the action, the mouse moving the wheel. And on the bottom right, you see the distribution of these neurons overlaid on the map that I show you on the previous slides. And so I think the little bit of neuroscience that I can give you now is that there's a tremendous amount of interactions and overlap in functional response across the whole brain. And so this means we actually need to study this tissue as a whole, as an integrated piece, which somehow contradicts a lot of the previous approach, where scientists were focusing on one area at a time. And so that makes our task a little more challenging. At the same time, amazing technology is coming together. So you see on the left here, this is a recent publication from a group in Rockefeller, where they are developing imaging technologies to record neurons across the mouse brain. So using two-photon imaging, so they're using a laser to scan neurons and record calcium responses. So whenever neurons emit an action potential, there's a massive amount of calcium influx, so channels opens and trigger. And so we use this as a proxy for recording the activity of neurons. And so over the last 20 years, and I contributed as well as other leaders in our fields, we have pushed new technologies to record more and more neurons, because I show you that this is really integrated. And so this group managed to record with a certain SNR up to 1 million neurons. So now we're getting to a place where, I told you there's about 100 million neurons. We're getting to a place where we kind of record a substantial amount of the network. But this technology are massive instruments. They're complicated optical pieces that are hard to disseminate. On the right, I'm showing you some of our platform at the Allen Institute, where we have dedicated engineers building integrated rigs that combine recording electrodes. So you see on the top, they're like electrodes going down into the mouse in the center here. And all of these arms are going inside the mouse brain. And there are multiple cameras that are watching the mouse at the same time to monitor what's going on. And so the challenge is whenever you have a system that is so tightly integrated, that is dealing with multimodal sensory input, that means you need to put together so many sensors to record what's going on. And that puts a lot of pressure on the experiments. So what are we doing to record this activity? And so I'm going to lean on our founders. So more than 50 years ago, UNIL and Visual led the characterization of visual responses in the cat. And I want you to notice, so these are Nobel Prize winners, in the background, all the equipments that was present. So it highlights the importance of technical integration and development for their field. And they were doing this together, but without like massive amount of team behind them. And so this has worked well for 50 years, but now we're moving with instruments that are so complicated that you need to have and the field is still operating this way in most of the lab across the world in terms of system neuroscience, Most PI are looking for the brilliant postdoc that is going to be able to do rig engineering, quality assurance metrics. So you need to check the data is good quality. You need to build system to check that once you have acquired the data, there's no data integrity issues. So we call this QC. That's a lot of work. And then you have to do experimental design. So you have to design the experiments so that it fulfills the questions you're trying to tackle. So now you have to do all of that. And now people are thinking we should also put the data open to the community because it's a tremendous value. Not only that, but also there are tremendous insight that can be gathered if other like modelers and computational neuroscientists can access that data and test existing theories. So open source is really important. So how do you do that while maintaining good work-life balance? Because I've done that for many years, and it's really getting out of reach for most individual students and postdocs. So we're trying to propose something that could actually help. So at this point, I think today, I think it's fair to say that most of the process is still closed. So we have you still see the thick lines. That's where we are today. But we have open access data publication, which is great. But in terms of sharing data and code, I didn't paint them in full orange because I think it's a challenging process. And some labs do it great. Some labs, I think they do their best to sort of share the data. But as you said, there's alignment incentive that needs to happen so that this can be facilitated. So I don't think we have fully achieved that. Okay, so to conclude this part, I think there's large potential for applying the cutting-edge techniques that I've shown you at scale. The brain is tightly integrated. It's a multimodal piece of tissue. And so I think as a result, we should be integrated with one another to tackle it. There should be a shared expertise in computational domain with like engineering and experimental labs so that they can focus on their domain and push the boundaries was possible. The modern methods are, I think, are complex, expensive, as I said, multidisciplinary, and they are not getting out of reach from academic lab. And on top of that, now you have to actually follow open source practices. It's just adding another ask from labs. So how can we do that? So now I'd like to leave neuroscience a little bit and talk about what other scientific fields have been doing. I think biology is a maturing field, but there are other places where this problem has been dealt with. So I have 3 examples to show you. And some of these are actually really familiar to you. So on the left, the CERN was a group of scientists that put together needs for a shared platform. And they tackle the problems many decades ago. They had to build teams in Geneva that dedicate their resources to building the platform, acquiring the data, and making it available. So much so that they invented the World Wide Web. So we just show you that they were tackling some of the challenges we're facing in biology. In the middle, this is something that really inspired us, partly because of connection at the Allen Institute with the observatory in Hawaii. So this is the pair of telescope that is exploring the night sky, and they are also a resource for the community. So they were built from nonprofit funds, and they're serving the field for free. But I want to spend a little more time on the one on the bottom right, which is about weather forecasting. There's an excellent book that I recommend everyone reads that tells that story. So it turns out, and I was surprised when I read it, it turns out that the weather forecasting field has been doing open science for more than 150 years. And the reason is that they have alignment of incentives. If you use weather measurements in a single country, you cannot predict the weather. You have to share because the weather in another country tell you what's going to happen in your country. And so as early as 1873, institutions across Europe went together to establish a system to share weather data. And at the time, this was sort of a time, like they were discovering sharing data through telegraph-based networks. So there's a story about this guy in US that could predict the next weather whenever the telegraph line was going down, because when it rains, the signal was dropping, the quality of the signal was dropping. And so very early on in the telegraph network, they learned weather forecasting was embedded. And in the following century, weather forecasting had to deal with AI problems. It's a very integrated system. The planet is highly nonlinear, and all of these pieces are tightly connected. And so you have to model the planet as a whole. And to me, that's really similar to the challenge we're facing in neuroscience. We have a really tightly integrated piece of tissue, and you need to take into account activity across the whole brain to understand its dynamic. And the nice thing about weather forecasting is that they had a really nice metric. So if they predict the weather well, and so they can measure how long we can predict in the future. And so very early on, they looked into building numerical models. And at the beginning, they didn't have the computational power, but over the years, they built those facilities. So I think we should look up for these folks on how to replicate, and they had to share infrastructure. So now they are building satellites to record this data. And it's obvious at this point, but this cannot be done by a single lab. They have dedicated expertise to build this and ship them in space. Now they operate the network of satellites and build models that are made available to the entire world every day. So it's incredibly impressive. So we were inspired by these models and tried to replicate some of those ideas. In 2012, Christoph Koch and Claire Reed put this piece out. And this was, I think, really pioneering some of these ideas and proposed that should build Observatory of the Mind. And so they went to Seattle and started this adventure. That's what convinced me to come. And all goals at this point, you have to put yourself back in 2012, where practicing of sharing data so openly were not so widespread, especially in neuroscience, maybe in weather forecasting, but in neuroscience, it was really much the beginning. And we had this crazy idea of acquiring the data and making it available to everyone prior to publications. Still to this day, this is not followed by everyone. So as you see, we have this dashed line now. So our goals was to collect, analyze, and publish data somehow in a more transparent way, and push out data set code and make publication open access, as well as make headways into sharing our protocols. This was our goal at the time. And so I'm going to try to show you what we have achieved along these goals. So in system neuroscience, an experiment is made of several steps. So you start from, typically, if you're dealing with mice, you start with transgenic mice. You have to do a surgery to give access to the tissue because the brain is incredibly well protected. And so we have to make openings in the skull. And that at the beginning, we started with imaging. So we wanted to use 2-photon microscopy to record neurons. And then we have to do some sort of mapping because you have to know where you are. So we use a technique called intrinsic signal imaging to locate individual brain areas. Then, as I showed you in one of the earlier slides, you have to do behavior training so that the mouse is performing a task that is important to your study. And then and do in vivo physiology. And then you have to take the brain out, reconstruct it in 3D, and map it to the coordinate system that I show you on the first slide. So this is a very typical workflow. It's like most labs currently, including many at EPFL, are doing this. And so we flesh it out and say, okay, what can we do to standardize this and streamline? So we built dedicated teams. So you see on top left, this is the surgical room. It looks like a factory. So there are and they're still running today. They probably surgery happening today at the Allen Institute. And so we have a team that is only doing a mouse surgery. It makes no sense to have someone that is doing computational neuroscience cannot be as good doing surgery. So we need to have specializations so that we can push the limits. And so but we did this for every step. So you see on the top right, this is an intrinsic imaging setup that is using the absorption of light by blood vessels to locate brain areas. On the bottom left, you see two examples of rigs using 2-photon imaging. And on the bottom right, this is our serial 2-photon tomography. So you have an image on the screen here, but it's showing you that like we took the brain out and we slice it slice by slice and reconstruct. So this system was built as a pipeline, and then we standardize with great details all of the coordinate system tools. So on the top left, you see a mouse, and this little blue piece is attached to the skull, and it gives us a coordinate system to position the mouse very precisely against the instruments. The neuroscience field has been using features on the skull to register a neural recording. So it's called BRAGMA. So it's actually also present in other mammals. There are intersections of points that we can reliably identify from mouse to mouse. And so this tools at the bottom left, this little pointy tool at the bottom, is used to precisely record that offset that the BRAGMA gives when you implant that little blue piece. And then on the right, you see that we have various versions of clamps that are attaching this head fixation across our pipeline. So this allows us to, this is about QA, quality assurance metrics. We check that every experiment is done with precisely positioned animals. So we did that, and over the years, we added more and more capabilities through this pipeline. So in 2017, we added NeuroPixel. So I show you a couple images. This was done using 2-photon imaging. So this allows you to record individual neurons in a 400 micron field of view. And then we added, so there's two main technologies to record neurons in mammals at this point, in rodents, humans using fMRI, but we're not using it here. So it's either calcium or voltage imaging or electrical recording. So neurons, whenever they fire a spike, they generate an electrical field that you can record with an electrode in the tissue. So this is the addition of electrical NeuroPixel recording. So you see in the panel C, we have 6 probes that are located inside the visual cortex of the mouse brain. This is the CAD that I show you in B. We saw it earlier. And then in D, you see individual neurons that are recorded across time. So every time there's a little dot, it means a cell was firing. And then you can measure this activity across many brain areas. So in the table in D, it's showing you at the top, you see the target. So these are names of visual areas in the mouse brain. And inside this table, this is the numbers of neurons that are recorded in each one of those locations. So with NeuroPixel recording, we have a higher yield because the probe is long and can record from many brain areas. So we typically record between a couple thousand cells per experiments. And this adds up as you do more and more mice, you increase your coverage. Later on in 2018, we added a behavior task. So you see, we are increasingly sophistication of this pipeline. So a behavior task is, in our case, was the detection of change. You see, in the first top rows is the mouse is watching a screen that is presented to it. And it's mostly passive viewing. We're presenting a stimulus and we don't ask the mouse to respond. So it's great to quantify visually responsive neurons, but if you want to study action, you need to have a behavior task. And so that's what we did. So we implemented a change of detection task that I'm going to show you here. So typically, neuroscience experiments have many different types of tasks. but this is a go no go experiment. It means you're shown a stimulus that is telling you go. So go for the reward. You're gonna get what the mice are water deprived, so they get a little bit of water when they go for a certain stimulus. And some of the times another stimulus is presented, and the mouse learn not to respond. So this is a very sort of standard type of class. And so you see on the right, so on the top left is the two kind of stimulus, the go and no-go stimuli. And on the right, you see the response of the mice to these stimuli. And so the little dots are little licks. So it learns to recognize the go trials from the no-go trials and go for it. And so this is really useful for us because then when we record the neuronal tissue, we can look for signatures of the stimulus that are associated with reward and the ones that are not associated with reward. So we duplicated this behavior task across both the imaging, the constant recording, and the NeuroPixel system. So that was sort of over the years. So our platform, and this was meant to be a community platform. So our platform sort of mature. So in 2016, we released the first characterization of the visual cortex with two-photon imaging. So 60,000 cells across many layers and cell types that you saw on some of the first slides. Later years, we increased that yield using NeuroPixel recordings, so 100,000 units. And then we had the behavior, so similar kind of yields, but with an additional behavior. And very importantly, all of our data was shared right when we finished collecting it. And so you can go to this link and right now on your phone and access this data, the entirety of it. So so in following on this data release, we started to analyze it ourselves because we were neuroscientists and interested in what was going on. And so these are two what we call platform publications that highlight the dataset and some of the insight that we gathered in this dataset. So on the left, you see the different types of stimuli that we presented to the mice. So there are gratings and so I'm sorry about the resolution. Drifting grating and static grating. So these are synthetic stimuli that a lot of neuroscientists, including the Hubel and Wiesel that I've shown you earlier, used. These locally sparse noise experiment stimuli are used to map the property of the cells and where they respond on the screen, because there's a visual map in the cortex. And then we also presented more natural stimuli like movies and image that are closer to nature. So we analyzed this data set and found that generally that the traditional models of the visual cortex is sometimes right, but most of the time wrong, which means that there are a large number of cells even in the first region of the visual cortex that do not follow the typical, let's say, simple responses. And then in the subsequent publication with the NeuroPixel data, what we saw was that so a lot of the field assume that there's a hierarchy of areas in the visual system. So, and this is coming from earlier work in cats or monkeys. So the idea is that, and this was actually extremely influential on AI models. So a lot of AI models are built with this idea of hierarchy. So you have a first area that responds to a small part of the visual field, and then as you go to the next area, it increases and it increases, and so you end up with visual features that are looking at the entire the visual field of your environment. So the idea is to have a hierarchy of areas that works this way. And what we found in the mouse is that there is a hierarchy, but it's rather shallow. So that means that most areas do have access to a lot of information that is far away, but there's still some sense of hierarchy. And this replicate what we saw with the connectivity. So we have analyzed the connectivity of the mouse brain and saw that the hierarchy is also shallower than assumed. These are how we share the protocols at this point. So we publish what we call wide technical papers, and this has not. I don't think these have been looked at as much as they should. So this is many pages long document that describing great details how we built our rigs. So we shared it, and then the CAD that I show you, we put a website at the Allen Institute to share that data. And so this is the sort of the 3D designs of the pieces that were involved in building the platform. So again, fulfilling on our goals of sharing a hardware. This is a very important part. So to share the data with the community, we built what we call the Allen Software Developmental Kit. So it's a Python library that gives you access to the data directly in the cloud. And we also built in some basic analysis functions that you can use to access data. And this has been used a lot since we released the dataset. I'm not going to show you the code, but you can imagine that this was a lot of work. So what happened? On the left here, I'm showing you a summary of the Keck Observatory, the one in Hawaii that I showed you earlier. And so this is a publication that they made to highlight the impact the platform had on the community. So you see the number of publications that are coming from Keck data. And so you see that it takes about 10 years to ramp up. And we saw the same thing. So on the right, this is a recent eLife that was put together by Saskia and Josh Siegel and Christoph Koch, where they looked at papers that have used our dataset in any way or form. So this includes some of our own work, but to a large extent, a lot of groups across the community. And so now there's and it's growing. There's more than 100 publications built on our dataset. For me, that's and I'm not an author on the vast majority of it. So this is for me. This is I think we are all very proud of this achievement. So this happened essentially. So you see, it turns out that we actually made full access to the analysis and the publication. And so some groups use our data to publish. So that meant that we really sort of opened it up. The way we collect data is still done in-house and was done for internal projects. So that meant that the research question and the design of the experiment was still close. But we succeeded in sharing CAD, protocols to some extent. I don't think we shared the protocols as much detail as we could have. And so I'm going to talk to that later on in the talk. But we had great success in sharing data, code, and publications. Okay, so this was in 2018, 19, and Christoph Koch, when he joined the institute, had really sort of this long-term view that we could change the way our fields operate entirely. And so this is what OpenScope is about. But what about just removing that little dashed line and collect data openly? So that means giving access to external teams for designing experiments and pushing them to a platform, the same way as astronomy operates. This is a simple idea, but it's against a lot of the way currently the field operates. So you have to evolve mentalities and the culture in neuroscience. So I think it's harder than it seems. So our goals were to maximize the impact of a platform and facilitate a community. So because any observatory is a community of labs across the world. And we also wanted to help develop the ecosystem. So I don't think all the pieces that an observatory rely on has to be built at the observatory. It has to be a decentralized ecosystem, otherwise it's not sustainable. And we have been going through two phases. So initially we were Allen funded. So the institute really believed in pushing this, and we are all thankful for that. But eventually we obtained, and this was really important to me, we obtained community funding to continue this effort, because this validates the model. It means that our peers think it's important. This is how we select projects. So now we're talking about receiving ideas and experimental projects that are deployed on the observatory at the Allen Institute. The experiment are executed, and then the data is shared with the external teams. So it's a very different way to operate. So how do we select projects? We decided on a two-phase process, a letter of intent. So that means a shorter document that you have to submit. And then a full proposal stage, so it's a 6-page long document, followed by external review. So it's really to align incentives. You need for this to be judged by your peers, not by people that have over interest. And so we decided to, and I'm so thankful for the external reviewers that are doing this review. And so they're volunteering time to review proposal. This is the same way as other fields operate. And then we have a steering committee that meets every January to finalize the selection, of which Mackenzie Mattis is a part of. I want to highlight that the deadline for the next call is next week. And so if any of you are interested, you can still submit projects at this website. So where are projects coming from? So we've been operating this for a couple of years now, and you see that they're coming from all over. So we had project coming a lot in the US, distributed from many institutions, from Berkeley, Princeton's, Wisconsin, UCSD in San Diego, as well as Canadian, McGill, York, as well as British Columbia this year, but also Europe. So this year we're running a project with an Israeli team, as well as 2 German teams. We also finalized a project with now a Korean team. So Yeyoung Shin proposed a project with Iloa Disnik, and she was in Berkeley, and then move on to South Korea. So this is really now an international platform. So I'm going to highlight 2 example projects. I can't speak to all of the 12 projects that we've run. So on the left, this is a project on predictive coding, and I'm going to speak a little more about predictive coding in the later part of the presentation. So the idea is fundamentally, a lot of people think that the brain is building a models of the world. So you're sort of exploring the environment, and you compare your expectation of the world with the input that is coming in. And so this team wanted to test some of this idea. And especially they were interested in the role of dendrites in predicting the world. So in AI, a lot of the models have like a summa, and then linear inputs that are summed. So you sum inputs, you compare it to a threshold, and you pass it to an activation function, or versa, depending on the model, and you have an offset. But there's very little non-linearity except for the activation function in AI. The real neurons don't work this way. They have really long dendrites that are themselves nonlinear. So people think of individual neurons as equivalent to a multi-layer network. And so there's a lot of research ongoing about the role of dendrites in brain computation. And so they were interested in looking at whether the dendrite contributed to encoding unexpected features in the environment. And to some extent, they saw that indeed the dendrites build up response to unexpected features differently than the soma in the same cell. In different cells, but we're running a follow-up with them on that. And so, and on the right, this is a project I mentioned. This project from Yeyoung Shin, where she used illusion. So you see on A, it's a famous human illusion. So I'm sure most of you in the audience are perceiving a triangle in this with this free object, and this triangle doesn't exist. It's your brain that is creating this object. And it turns out that actually mice perceive illusion. This was debated for many years, and Yeyoung Sheen as well as others show it's actually the case. But this kind of stimuli are really helpful because you can turn on and off the presence of the object and then learn and study how the cortex might be created by binding features. So you have features that are responding to the little Pac-Man on the top right. So the 4 Pac-Mans. And so when they're aligned, they must be processed in the brain that are sort of connecting the responses of those cells responding to these different parts of the stimuli. And so she indeed showed that that's the case. You can look up at a publication. It's currently in review. Okay, so I show you 2 projects, and but we also, as part of the OpenScope, committed to release the data to the community at large. All right, 50 minutes. So this is so to do that, we use a platform. So I told you that we didn't want to reinvent the wheel, and so the community has built a platform called Dandy. This is a team at MIT that is leading this. It's a community data sharing platform, and so we decided to use it instead of build our own. And so you see here that in red, some of his data set that I highlighted, like the Illusion project, are marked down as embargoed. So as part of our project, we to ease the transition, because the field still expect to be able to analyze data, because there's a lot. I mean, individual labs, if they submit a project to us, they could be concerned that if we push the data out immediately, they'll be scooped on their own idea by a team from the outside that analyze the data. And so we decided to choose a middle path and embargo the data for 1 year and then release it later on. So this was sort of a compromise to help move the field into this model. And so in February, we actually released the data. So for this project that I mentioned. And so we organized, this is just to show you how we did it, how we sort of facilitate open science. So we organized an online webinar with individual teams, and they spent 40 minutes to present their data set and highlight their analysis. And we also sort of incentivized collaborations and gave us a link to a Slack channel where people can talk to the team and facilitate collaboration if you are interested in a particular data set. So this is something really recent that we started to do. But you can go there and watch our data release webinar. Okay, so now I've shown you that we share the data, and so now it's on this platform. It's terabytes of data that you can access, individual neural activity in response to the stimuli that the external team designed. So how do you facilitate analysis? Because this data set are complex, and so you need Python code to facilitate that. And so we design what we call the OpenScope data book. And so this way, again, we don't want to reinvent the wheel. So we are using a data format called NWB files, which was designed by a team in Berkeley, storing those files on Dandy. And then we created in collaboration with a lot of labs across the community that Jupyter book. So it's not a Jupyter notebook. It's organized as a book. So you can go there. It has, and because we had all of this data available online, we thought this was an opportunity to make a really great educational resource. And so we put the effort to do that. And so there's in this data book now, there's like I want to say like 30 notebooks now that highlight some of the basics analysis that every neuroscience lab is dealing with, like building receptor field or understanding what the cell is responding to, what are statistical tests that you use to show that. And so we do that with the text, the code, and access to the data. So you can replicate every of this notebook by clicking on the little rocket button at the top, and it brings you to Google Colab. So it's really easy to use, and it has references to the papers that we use. So it becomes this really nice community foundation set of notebooks that have everything embedded into it, from code, data, and references. So it's essentially a paper. And we thought, I know you talked about this yesterday, but we thought, why not try to a new very open publication model? So Jupyter Book are really easy to host on GitHub. And so we essentially decided not to publish this in journals and push it on GitHub only. And because this is a really nice ecosystem, we also made a system to have authorship automatically attributed on merge. So that means anybody that makes a PR on the repo gets credits by when the PR is merged. So we're using the GitHub, this is software engineering tools, pull request system to manage authorship. And then we reach out to a couple community members for reviewing this work. And so they were graceful and they did. And so we give them authorship as well. And so it becomes this really nice publication that has code, data, and text all embedded on the repository. And the nice thing about this is you can version this now. So it's a paper that you can version online, and you can mine to DOI via Zenodo. So this is entirely citable. So it's an experiment. We're going to see if it picks up, but it's a completely it's not using the traditional publication system, but it's entirely self-contained. And we decided to use this to test this because this is like a great educational resource, right? You're learning how to use neuroscience data with these notebooks, and so this is a great way to embark the new generations on really sort of strong open source practices. Okay, so in addition to sort of sharing our code and data this way, we also organize workshops. So this is to facilitate a community building in our community. And so every year now we organize online technical workshop at the Institute. And I've been surprised. People from all over the world sort of were able to find the funds to come, and we show everything from start to finish. So the same way as an observatory should be a hub, a technical hub to share access. So this is how we really push the needle of sharing our process. And so you could apply, anybody in the audience could apply in February and come at the Allen and see how we acquired the data and bring it back home. Okay, and I mentioned that this is sort of ongoing. We are now pushing our protocol documents to protocols.io. So this is a reflection of our internal standardized operating procedure that we had already built in-house, and push it also with DOI. So now people can reference boost protocols and re-implement them in their own labs. So we're not completely finished with this, but I just want to highlight that we are going this direction as well. The other things that is going on the Allen is we are developing a new system based on Code Oceans to do analysis entirely in the open. So some of you might be familiar with Code Oceans. It's an Israeli company. They have a partnership with Nature journals to push capsules attached to papers. So it allows to replicate computational environment. Reproducing an analysis is an ongoing challenge, right? Because you have to bind data and code and environment. And so we are migrating our entire system to this ecosystem. So, and I think in the coming year, this is really going to come together, and you'll see that we envision a future where the processing pipeline that we are using to manage the data will be reusable by external lab from start to finished, because it will be entirely in the cloud. Okay, so this happened through the OpenScope project so far. So you can see that we made the collection of data now completely open access, so people can submit projects. And I think I put a star on open access publication because I think this is like unexpected experiments, and we'll see how far it's going to go. But I think we've sort of show a way to do open science in the neuroscience experiment. But what about this? Could we just open the whole thing? Do we need to be closed? So could we do a process where the question is defined by the community openly? Like, do we need individual labs to secretly put together questions because we're afraid of everyone, and then do their experimental design for a couple of years and get scooped eventually in the end because everybody had the same idea in some ways? Is there a way, is there a path so we could just open the whole thing? like do science. I like also SAS categories like to call it science in the open. And this is really far from what the field is doing. And we thought that the OpenScope project could be a way to really try to push for that approach. I think it's important because it could show a very inclusive process. Also show the all of the future generation that science can be done extremely openly. So we tried this twice. Last year we started a process, and so we created a forum where people could come, and I was going to be interacting with them and facilitating team formation in a very open way. It completely failed for some reason, but I think I understand now. But this was not, there was no targeted question. It was just like, okay, guys, if you want to discuss an idea, we'll there, and then we'll make it in the open. But that didn't work. I think people were confused. You know, like it's so different from the current way we design experiment that it needed a little more, I think, a little more leadership. And so this year we tried again with the. I want to so again thank Mackenzie Mattis and Sandra Gosh in the steering committee as well. Nick Steinmetz, who really are supporting of this, doing science in the open. And so this year we decided to select a very specific question and try to design an experiment with the community around that question. And this is around ideas of predictive processing. So we saw that over the years, many projects that were coming to us were related to this theme. And so we felt, if everybody's interested in this question, why don't we design an experiment with everyone? And so here I'm highlighting a diagram that we made in the last few months. So it's showing you sort of predictive coding is this theory pushed by a lot of labs over the last 20 years, including Rajesh Rao in 1999. It's a very influential paper that a lot of people, because if you put a microscope in the brain, the first thing you notice is every time you clap, the whole thing light up. And so this means that there's a tremendous amount of responses to unexpected events in the cortex. And so a lot of folks were trying to find, let's say, computational layers underneath to explain those responses. And so predictive coding emerged. And the idea is that in each brain area, there are two types of neurons. There are neurons that predict the outside world and neurons that are specific to errors, like when there are errors to those predictions. And you can look up the references along those teams, but they have, folks have shown that you can train models that operate this way with specific units dedicated to that. And especially if it's hierarchical, like the idea is that the error signal is sent to the next area instead of the whole prediction, so that it's sort of a little more efficient. Over the years, there has been various implementation of predictive processing ideas. And recently, a lot of people have been interested in the role of dendrites. And so they think that maybe dendrites are actually where the error response is located. But we still need a lot of data to sort of educate between the models. So we decided, why don't we design an experiment around these ideas? And as I said, this is an example OpenScope project that is the first time I'm showing you this, but that tested some of these ideas. And so they designed an experiment where you see a sequence of image, A, B, C, D, A, B, C, X. So the X is the unexpected image in the sequence. And they were looking for signature of neurons responding to this unexpected image. And they saw indeed that there is a distribution of responses across the hierarchy. So supporting some of these ideas. And then this is the paper I already mentioned that was looking at dendrites. So we already had a core group of people that had analyzed our data and looking into this question. So we thought, let's organize a workshop. And so in August last month, we submitted a proposal to a computational neuroscience conference called CCN that happened every year. It's around, sometimes it comes to Europe, sometimes in US. This was in MIT. And the program of the workshop was design an experiment that the community needs. And so this actually started to work. There were a group of people that gathered around it. But the thing that I really highlighted what was encouraging for me was not the conference. It was the Google Doc. So we put a 60-page long document online with complete open access. Everybody can edit, put comments. And it turns out that research is a really complicated field. You need depth. And a conference is too short, I think. So what we found is that the highest quality interaction that we got were on this Google Doc, because people had the time to think and then reflect. So now there's more than 25 authors on this piece. It's really open, like everybody that contribute can be added as an author. The number of it for me, I've spent an hour a day for 2 months just managing the commons and helping shepherd that process. But it's really been fun. Like I've never seen my field operate this way, where we're really discussing and reviewing the community. So if you want, you can go there. It's still ongoing. So we're defining this experiment. And if you have interest in predictive coding, please chime in. Okay, so far we are there. I think we have shown that we can start to design an experiment in the open, but we haven't done it yet. So I want to say that that's why I put sort of fader colors. But I'm hoping that next year we have shown it. Okay, so to conclude, I hope I've shown you that we are operating as an observatory in neuroscience, and I've pushed the boundaries of what can be done. And I'm really excited about this Science in the Open conclusion. I think it will be a crowning achievement for the program. We've completed 12 projects. There's 4 more ongoing. I can't sort of, I don't have time to speak about all of them. But I think what we built is a foundational platform to study brain-wide during complex behavior. And in the context of AI, I think this is the foundations, and there are layers that needs to be built at top. Every biology is a field where the idea of causality is really hard to understand. Any neurons in the tissue receive 10,000 inputs, so it's really hard to say response is caused by this area or this neurons. So you really need integrated layers to study causality in biology. And I think the right approach is probably to build foundational platforms, such as one of these, and then acquire data set across the community that really tackle the right questions, and then build these models using AI on top of it with like stringent quality control so that everybody trusts the findings. And I think if we do this science in the open, it's going to move our field forward. So that's what has been driving me for the last decade.